her
Thanks,
session.
Anva. Now take a small journey from where we started our life. We all were very happy, lived in ocean, big ocean, single cell being, no problem of energy, lot of to conserve. So the system evolved to get rid of extra food, like Leishmania, Plasmodium, Trypanosome. And when protozoan became metazoan, then there was development of gut took place. And now food has to go to a complicated system. And the first glucose transporters known as sweets, which eventually we exported against the gradient in eukaryocyte developed from prokaryocyte, and ultimately there was further families divided into several subtypes. So right from the single cell to the multiple cell, we did not have mitochondria at that time. Mitochondria was incorporated into the cell, learned how to do the glycolysis, and this oxygen atmosphere was oxygen free, was anaerobic. Ultimately, it is the chloroplast which was synthesized into the botanical part, cyanobacteria, blue-green algae, and then oxygen was released. So we learned then with eukaryocyte how to conserve the oxygen and then form. So initially, we all developed co-transport system with sodium because sodium was more outside. So as the Claude Bernard said, middle interior, we had a pump to throw sodium out, and we also had to throw sugar as well. And you look at the distribution right from the conguloflagellate, porifera, plesiozoa, mollusk, annelids, arthropods, and ultimately human being, we all had a different level development of this particular glucose transporters, which are known as the GLUT, and SGLTs, which are the active transporters. This is very ancient family. The major function of these proteins is to carry out secondary active transport. Multifunctional proteins, diverse function. SGLT1 by evolution is the first glucose transporters, and there are approximately 450 members, 230 genes that share the common synpods. There are three superfamilies, and the major facilitator superfamily called MSS transporters, and newly identified sugar will eventually be transported what called sweet. What is sweet? This is a transport system, can be classified into 4, 2, 2, and 2 clades in plants and metazoa. They are unicellular eukaryocyte and also in prokaryotes. And we needed it because the moment we had a gut, the gut has to absorb, and the first encounter came with SGLT1 when taken to the capillaries, then to the circulation, then SGLT2. And if the first clone transporters within the large sodium solute mediates active glucose and galactose absorption and has a mutation. And due to its fundamental physiological significance, SGLT1 has been the focus of many functional and biophysical studies that have shaped our understanding of active transport. And this is the complex family. And look at on my left side, there are the different genes which are responsible from SGLT1, 2, 3, 4, and 6, and they are responsible for glucose, galactose, mannose, fructose, and chloroanisotol. The mollusks are the first one to start with, and it started right from the fish onward. And look at the complex, this is a very complex slide, but what I wish to talk to you that from archaea, bacteria, fungus, green algae, blue algae, every plant has this memory which is related to SGLT1. And this complex shows on the top, there are SGLT4, 5, and 1 distributed on the Homo sapien, that's the human being, and below there are the chordates, then there are the amphibians and the avians. I'll talk quickly one or two. There are two models of sugar absorption in invertebrate marine animals, and these are the primarily SGLT1 mediated, followed by GLUT4 and GLUT5. Then crabs, again its presence excessive SGLT in the crustaceans, and these are the hepatopancreas brush border cell from lobster, where they regulate the absorption. And SGLT4 is present in hepatopancreas member of the lobster. Mucosal cell of the goldfish also contains SGLT1, and the border contains GLUT2 and GLUT3. And this is what we have, this have shown the presence of the GLUT2 on the lamina propria of the intestine and also SGLT2. Tadpole, and why this is important, why amphibians I have put, because as the animal moved from the aquatic became terrestrial, the pronephros became mesonephros, the mesonephros became metanephros. So you had a long tube, the collecting system, the proximal tubule, S1, S2, S3, and then loop of the Henle, and that's the collective tubule. And this led to the increased absorption area, surface area. Why? That's the reason why SGLT1 became only 10% at the S3 segment, 90% became at the proximal segment. So tadpole and frog also has SGLT1 primarily, and then looking at the nephastine, again, then you have a system of a very complex system of GLUT2, 3, 4, and 5 with SGLT1 only. This is the reptiles, and again reptiles have the similar distribution. Look at the bottom, glucose transporter SGLT1 metabolism. You do not see SGLT2 as yet. From the birds onward, avian, proceeding from the avian onwards, look at the bottom, SGLT2 protein started appearing with GLUT2, and now we have this complex family of the 5 SGLTs and the GLUT10 and the sweet family. So looking at the different genes which are responsible and present there on the intestine, trachea, kidney, heart and brain. So most of the organs have shown this distribution. And this is what the, look at on the right side, hardly there's any organ which doesn't express SGLT2. So when you innovate a molecule, you have to take into the cognizance that the transporters at other part of the body equally play significant role. This you have seen time and again, so I'll quickly take to the endogenous production of the second part, that is the gluconeogenesis. Why this complex problem has occurred? Because as the CKD is becoming more prevalent, over the time, you would also see that these molecules, they tend to fail the response. And you will be surprised to know that 40 to 50% of the endogenous gluconeogenesis occurs under stress, acidosis, fasting, and makes second most important organ as the kidney. And in the kidney, gluconeogenesis is mainly performed in the renal cortex, in the proximal tubule, and only the segment expressing the required machinery for this process. This allows synthesis of glucose 6-phosphatase. It's a reverse order, non-hexose precursor, and 11 enzymatic reaction. And therefore, in kidney, the main precursor for gluconeogenesis under physiological condition is lactate, which is 50%, and 20% is the glutamine, and 10% is the glycerol. So what's the advantage of this biochemistry? Advantage is the reverse part. It is responsible for about half of the total gluconeogenesis. Insulin and catecholamine and other hormone is being regulated and takes place in the cortex, and medulla is the principal producing organ for the other enzyme. Look at my right side, and this is the reversal of the EM pathway, Embden-Meyerhof pathway, which was responsible for the glycolysis down from the pyruvate to the synthesis of the glucose. And also, this is because of the hypoglycemia. One of the reasons of evolving newer molecules when you use them in chronic kidney disease is the presence of the hypoglycemia. And the reason being, you have extreme glomerulonephritis distorting the cortex. They cannot do renal gluconeogenesis. If the cortex is very healthy, like acute interstitial nephritis, chronic interstitial nephritis, pyelonephritis, polycystic kidney disease, hardly then you get hypoglycemia when they are associated with diabetes mellitus. So look at this model and look at the CKD1, CKD2, CKD3, different stages of chronic kidney disease, and look at the gluconeogenesis. So as the chronic kidney disease advances, the gluconeogenesis also goes down. Therefore, the glycolysis goes up, and this is one of the limiting factor in evolving newer molecule. Coming to the SGLT2 inhibitors, and that's what the nephrology has a role to play, that on one side you have a catabolism, you use very high dose of SGLT2 inhibitor, you produce almost fasting-like state, you increase the ketone bodies. On the second part, it is taken as a compensatory mechanism for the anabolism, which is good for the heart, ketone bodies is good for the heart, and increase glycogen and protein synthesis. So looking at the deep into the cell to find mechanism of kidney protection by SGLT2 inhibitor, these important observations offer insight how SGLT2 inhibitors may protect the kidney that take the evidence beyond investigation of nephron solute transport. All these molecules were primarily for the treatment of the diabetes. Now it has become drug of choice for the nephrologist. Now it is becoming drug of the cardiologist. Therefore, the patients who received SGLT2 inhibitor could conceivably be either higher or lower risk for DKD based upon the clinical decision made outside this study. And therefore, although the SGLT2 inhibitors appear to have class effect for protecting the kidney that is largely dose independent, biases may also exist due to use of various SGLT2 inhibitors in different doses. This is what have been shown with the marker, and look at the cortex where you get excess of the DAPA which binds, which is SGLT2 inhibitor. This is valuable slide when you look at the 24-hour protein, and this is what the action which is not because of the glucose lowering property or sodium hydrogen on exchanger. When you have a 24-hour uridine 10,000, and look at the post-treatment, and look at the reduction. But when it is less, it is very, very minimal, almost equal pre-treatment and post-treatment. And therefore, probably, the early use of SGLT2 inhibitor to reduce the proteinuria is more scientific rather than using at the late time. Now look at the CKD distribution of all the 4-gliflozin, and look at here the hazard ratio, and look at the eGFR distribution. This is 25/75, 20/90. Higher is the GFR, better is the result. So dapagliflozin, this is 44, this is 28, this is 44 again with canagliflozin. But look at the bottom, the cardiovascular outcome and the hazard ratio. Maximum here, that is 31, this is 23, and this is only 17. Meaning by, if you can block efficiently SGLT2 inhibitor at the cortical level, when there is good eGFR, probably the cardiovascular protection will be much better than at the later stage. And this is the last few slides, 1 or 2 slides. Looking at the value on my right side, on one axis, this is 60 eGFR, and you start using SGLT2 right over here. You add number of years, almost 11 years. So using SGLT2 inhibitor in the beginning would give you extra lease of life of 11 to 12 years rather than starting at the low eGFR of 30. And that was the purpose of choosing this topic. How gluconeogenesis contributes into the selection of SGLT2 inhibitor as a potential molecule. Therefore, to summarize, SGLT2 are most ancient gene across metazoans to human being. Sodium glucose co-transporters has 220 or more members in animal and bacterial cell. There are 11 human genes expressed in the tissue, and sugar will eventually be exported. Sweet, which is known as sweet, is a recent characterized families, but it is in evolution, this is one of the older one. And also, across the animal kingdom, from pathogenic protozoan to mollusk, arthropod, amphibians, reptiles, avians, human beings, they play active role in glucose homeostasis. SGLT1 was first to appear in the gut, 90%, followed by SGLT2, and therefore now we are looking for the molecule. The present SGLT2 inhibitor inhibits only 40% in proximal tubule. Remaining is taken care by SGLT1 at the ST3 segment. Renal gluconeogenesis contributes 40 to 50% of body requirement, and DKD and CKD pharmacodynamics SGLT1 inhibitor, SGLT1 and 2 targeting has significant potential given their expression profile, yet dual inhibitors may offer a combination of benefit for type 2 diabetes management due to their effect on UG and incretin hormone, and understanding of cluster and phenotype in DKD may have a role to play in the therapy. Thank you very much. I finished in time.